Well, I'd like to welcome everyone back to the Gambier Symposium. My name is Jason Lee, and I'm the Deputy Director of the Molecular Imaging Program here at Stanford. My connection with Sam is a little different from others in that I've only met with him since 2019 when I came to Stanford from UCLA. But my background at UCLA actually suggests that I've actually known Sam for quite a long time. He's definitely still a powerhouse in our imaging program and field across different universities. And to go to the analogy about the train, I would say that I haven't hopped on the train until 2019, but way before that, I was running after the train trying to catch up with Sam and all of his other colleagues. So it's a privilege for me to be able to moderate this session. And our speaker for the session is Dr. Carolyn Anderson. Dr. Anderson is the Simone Elbrock Professor of Medicinal Chemistry and Professor of Radiology at the University of Missouri. Her lab focuses on the performance of and design, synthesis, and biological evaluation of novel radiometal-based radiopharmaceuticals for diagnostic imaging and targeted radiotherapy of cancer and other diseases. A current focus of her research lab is the development of imaging agents for upregulated receptors on immune cells that are involved in inflammation and primary tumor growth and cancer metastasis and sickle cell disease. So without further ado, Dr. Anderson.
Great. Well, thank you so much, Jason, for the great introduction. I'm going to try and share here. Hopefully that looks okay. So yeah, what an honor it is to be here. I'm so thrilled to be at this symposium. And my connection with Sam was, he was the chair of a study section that I did for many years with him. And that was in the Cancer Prevention and Research Institute of Texas. And Sam was phenomenal. Our study section covered every topic imaginable. We were sort of the other category. And so imaging or biomarkers or looking at various types of technology. But Sam would, during our little breaks, or if we were in between grants, Sam would always bring up some article or something he read that was like future-driven technologies that he would always like tell us all about. So it was just really fun to work with him for all those years. And you know, we really miss him tremendously. And so in the vein of my talk, I'm actually going to focus, I'm going to focus on a collaboration I have with a company, and I am a member of their scientific advisory board. This is Lumiphore, because I am also involved with one of the companies Sam founded called Early. So I'm really excited about that as well. But today I'm going to talk about some of the work we just started at the University of Missouri and working with Lumiphore. And so in the outline of my talk, I'm going to talk a little bit about, because you're probably wondering what's going on at University of Missouri. I'm in the smack middle of the state, small college town. But there's really a lot going on here, and we hope to be, and we are actually a hub for a lot of Theranostics research. So I'm going to talk about chelator chemistry and focus on one particular chelator that Lumiphore developed, which is somewhat of a universal chelator for metals that are +4 or +3 charged. And I'm going to tell you about two molecules that we have looked at as conjugates of this particular chelator. So in terms of the University of Missouri, I don't know how much everybody here knows about it, but we have a whole bench to bedside approach. And you may not be aware, but we have the largest research reactor in the country. It's called the Missouri University Research Reactor, or MUR. And MUR produces 5 radionuclides that are used clinically throughout the country, including we produce Tech-99M, or the generator, molybdenum-99 for that. We produce I-131 and also lutetium-177, among a couple others. At MU, we also have an NIH-funded mutant mouse resource, and we have a swine center as well. We have a College of Veterinary Medicine, so we can do translational studies in companion animals. And then we also have a brand new translational health facility called Next Gen Precision Health. And but what I'm going to focus on today is what work we are doing in a center that I now direct called the Molecular Imaging and Theranostics Center. And we are located directly across the street from MER, so that we get the radionuclides from MER, basically walked across the street to our center, where we are now doing most of the basic chemistry and the preclinical imaging. And I co-direct this center with Jeff Smith. I believe I'm the first talk here today in Theranostics, so just to give you a little brief introduction, that this is not a new technology. Theranostics have been around for a very long time. I consider I-131 sodium iodide is a theranostic because you can do both diagnostic imaging and treat cancer with it. But the latest therapeutic radiopharmaceutical to be FDA approved is lutetium-177 PSMA 617 for imaging metastatic or therapeutic for imaging and therapy of metastatic prostate cancer. This was FDA approved on March 23rd of this year. Its trade name is Pluvicto. And there's a companion diagnostic, which is gallium-68 PSMA 11. So these are actually two somewhat different compounds, same targeting molecule, but two different chelators here for gallium-68 and lutetium-177. This agent is called Locomets, and this was FDA approved in December of last year. And it's worth pointing out that the lutetium-177 for Pluvicto is being produced at MER, and pretty soon we'll be producing the no carrier added lutetium. Right now, it's carrier added lutetium that is being used in this radiopharmaceutical. So I like to point out that my research, we attempt to cover the whole cycle of molecular imaging and theranostics development. So the radionuclide production happens at MER. So we get radionuclides fresh off the reactor. We do all the chemical synthesis here, the chelator synthesis, and the radio labeling. We do all the in vitro assays that to test our agents to make sure that they still bind to the molecular target of interest. And then this is followed by preclinical imaging, which we then validate ex vivo to make sure that what we saw in the preclinical studies is we're truly hitting the target that we intended. At MU, we have it's a bit little bit more unusual in that we can go into larger animals that include companion dogs, and we can also look at swine models. And ultimately, we want to do the first in human imaging, and this can be done at NextGen, where we have a research dedicated PET-CT scanner. Oftentimes, what we see in the human images doesn't look quite as nice as the mouse, and so we have to repeat this cycle again. And what the center that I direct and the building that I'm in, we really focus on the chemistry all the way up to the preclinical imaging, and then we collaborate with others here to do the rest. So the radionuclides I'm going to be talking about today are, we have a diagnostic radionuclide, zirconium-89, which has a half-life of about 3 days. So this is good for, especially good for protein labeling and maybe nanoparticles as well. And then lutetium-177, which I mentioned, which we produce across the street at MER. It's used in FDA-approved products. It has a half-life of about 7 days, and it also has imaging gamma photons for SPECT imaging. And a new radionuclide that is it's being tested right now in Europe in a few clinical trials, but it's pretty new to the United States, which is terbium-161. And the benefit over lutetium-177, they are pretty equal in all ways, except that there's a significant amount of low energy electrons. So if we can deliver the terbium-161 inside of tumor cells, we have a better possibility of doing cell killing due to these low energy electrons. And there's also some gamma emissions that are useful for SPECT imaging. So terbium is being produced under a Department of Energy grant that Heather Hankins, who's one of my colleagues here at MU, is the PI of, and her student Patrick Piccolo. So the terbium-161 is actually produced in a no carrier added production starting with gadolinium-160. And so this way we can produce higher activity terbium-161. So the chelator that we've been looking at is called LUMI-804, and this is an octadentate macrocyclic compound. It's based on these 4-hydroxypyridine 1, it's a 1,2 HOPO coordinating units here. And this was developed by Darren Magda and David Tatum at Lumi4. It's a company that was started by Ken Raymond at UC Berkeley. And what's really nice about this octadentate chelator is it's not great for some of the transition metals like iron or zinc or copper. And so it really does prefer these larger atoms such as zirconium-4 and some of the lanthanides, as well as thorium, which I'm not going to talk about here today, but it has also been tested for that radionuclide at Washington University. And so we've previously published with this chelator looking at lutetium and zirconium in looking at antibodies. And I'm going to tell you some of the more recent work that we've been doing with this agent. So Darren and David have done all the cold chemistry, and they've shown that this compound, it metallates at very low temperatures in about 30 minutes, and it has a very high kinetic stability when you challenge this with high concentrations of DTPA. So one of the compounds that we are using as a model is a compound that I've developed in my lab, and we've looked at it for many years now. And the target is an integrin alpha-4 beta-1, or very late antigen 4, which is an integrin, and it's expressed on melanoma, lymphoma, and multiple myeloma, as well as various immune cell types. It recognizes VCAM1 and fibronectin as its natural ligands, and it plays an important role in tumor growth, angiogenesis, and metastasis by promoting adhesion and migration of cancer cells as well as immune cells. And the compound that we've been looking at actually binds to the high affinity state of this particular integrin. And this compound is LOP2A that was developed by Kit Lamb at UC Davis. And what we've done is we've changed the linkage here to a PEG4 to give it some more water solubility. And basically you can put almost anything here in this R group here, could be dyes or chelators. And several years ago now, we published, we looked at 2 different chelators for copper 64, with the crossbridge chelator actually being the best chelator for this particular agent. And this copper 64 crossbridge chelator LOP2A is now being tested in 2 different clinical trials, one at Washington University in St. Louis in multiple myeloma. And we have a new clinical trial in the new NIH grant to look at copper LOP2A in terms of imaging vaso-occlusive episodes in sickle cell disease. We've been testing this in just when we modify the compound, we've been testing it in a mouse model of melanoma for simplicity's sake. So the LUMI-804 LOP2A, it readily labels with lutetium, terbium, and zirconium, all at room temperature in under 30 minutes and very high purity. So this particular agent, very easy to work with, very easy to label. And this can be compared to some of the DOTA conjugates, which we require high temperature. And I'm going to talk about that in a little bit. So this is just an example of the terbium-161 LUMI-804 LOP2A. We show that it retains high binding affinity to the VLA4 integrin in this melanoma cell line. And we've tested all of these compounds, and they all have very high affinity to the VLA4 receptor with lutetium, zirconium, or terbium. At University of Pittsburgh, we did some imaging studies with the zirconium-89, and we see very high tumor to non-tumor ratios with the zirconium agent that we can then block with cold compound, and this is at 4 hours. And we also see retention in the tumor out to 24 hours. This is the biodistribution data. And these are data over here that just showed we did the studies in both male and female mice. And there's really no difference between the male and the female mice in this particular model. So now I'm going to pivot a little bit to, we have a small business grant and SBIR from NIH, so that Lumiphore holds the grant. We're also collaborating with Imaginab, and we're looking at their this is an anti-mouse PSMA mini-body, which they have an anti-human as well. And so what we wanted to show in this, with this SBIR research, is that Lumi-804 would be a great chelator for lutetium, zirconium, as well as down the road, thorium-227 for alpha therapy. And so we did a comparison, a direct comparison with DOTA, which is the standard chelator that's used for lutetium. And just to show you in terms of the radio labeling, and people do use DOTA for radio labeling antibodies with lutetium. And we have to heat it to 37. We can't really heat it much above that without destroying the protein. It requires 2 hours of labeling, and we're not getting a complete labeling. So right here is, this is actually, we had DTPA to try and remove this lutetium, free lutetium. So we're getting only about 90% labeling. Whereas when we do this with a Lumi-804, it labels in room temperature at under 30 minutes and requires no purification whatsoever. But we are able to make the lutetium DOTA agent. And we did a biodistribution study and compared the DOTA versus the Lumi-804. This is a lot of data, so I'll just point out a few things. One thing we noticed with the DOTA is that we actually do get higher tumor uptake with the DOTA conjugate than we do with the Lumi-804. However, we are getting also higher uptake in the non-target tissues that include the liver, spleen, and kidney. And since we're targeting PSMA, we also looked at the salivary and lacrimal glands, and also we looked at bone. And what we see here, bone is a really good indication of stability, and we're seeing high bone uptake with the DOTA chelator. We're also seeing higher uptake in the salivary and lacrimal glands, and we're seeing clearance out of these tissues, which tend to be areas where we see toxicity with some of the PSMA targeted agents. So in that respect, and another way to look at it, this is just looking at the tumor uptake versus over time. Again, lower uptake in the two than we see with the DOTA, but we also see here that we see a lot higher uptake in the kidney and liver that doesn't clear out as well than we do with the Lumi-804 chelator. And then also, again, we see clearance out of these non-target tissues where we see retention with the DOTA conjugate. And then to look at the tumor to non-tumor ratios, we see much higher tumor to muscle, tumor to blood, even higher tumor to kidney and liver than we do with the DOTA conjugate. So in that vein, we do think that the Lumi-804 is going to be a superior chelator and more versatile that we can look at with, we haven't done the zirconium imaging yet, but those experiments are underway. So to summarize, I told you about a new chelator, Lumi-804, which is very versatile. We can use it for radio labeling the lanthanides for beta therapy, zirconium-4 for PET imaging, and also down the road, thorium-227 for alpha targeting. We've shown that with LOP2A, all these agents bind just fine to the receptor, and we got very nice imaging with the zirconium-89 and PET. And then with the mini body, which is, I think, really where we're going to see that this chelator is going to be great with is either intact antibodies or engineered antibodies. We show that the Lumi-804 has major advantages over the DOTA chelator. And down the road, we're going to be finishing up the studies with zirconium and thorium with the antibody, and hopefully bring this eventually into the humanized antibodies. So with that, I'd just like to thank the people that I get to work with every day and my collaborators at Lumiphore and Ken Raymond, who really, I think initially came up with this chelator design. And I'd like to thank you all for your attention. Happy to take questions.
Thank you so much, Dr. Anderson. That was a fantastic set of results that you have going. that Lumiphore increase in biodistribution and tumor uptake is fantastic. We look forward to it. So the floor is open up to questions. Please feel free to type your questions into Slido. So perhaps I can start with a question that I had regarding site-specific radiolabeling. Do you think for antibodies, how important is it for us to actually do site-specific labeling as opposed to pan labeling?
I mean, Mike's, I think site-specific labeling is going to be more important for some of the engineered constructs like the mini bodies and the diabodies, maybe over the intact antibodies. You know, just some of the results that I've seen and experience that we've had, the non-specific conjugation, especially on intact antibodies, doesn't seem to make a huge difference, but I think it's also going to be very antibody dependent. But I think that would be a nice way to also look at this. I think this chelator is pretty versatile with respect to how we want to link it to constructs as well. Great.
We have one question from Noeen. Noeen says, good and interesting presentation. I just have one question. Any comment on percent purity of radioisotope terbium-161 versus yield or on radioisotope purification?
Yeah, I didn't actually show the data that we have. So right now we're producing pretty low amounts of the terbium-161. So we're only producing about 25 to 30 millicuries. And so there's going to be inherently some impurities in this low amount. And we produce even carrier added lutetium-177 because it's done commercially and for routine clinical use, curie amounts are being produced. But I have to say that with the DOTA chelator, we definitely see more interference with traditional contaminants such as iron or maybe even copper and zinc than we do with the Lumi-804. So we're actually able to get higher molar activities with the same Lumi-804 constructs that we get with the DOTA constructs. And we've got a whole study. I had a master's student who finished a project on that we hope to get published soon to just show that we think the Lumi-804 chelator is also going to be able to achieve higher molar activities than some of the traditional chelators that we use.
So related to that, Jimmy Chen says, wonderful talk. Will Lumi-804 chelator change the affinity of the antibody to the target?
It definitely didn't change the affinity for the small molecule LOP2A. And we did do or actually Lumi-4, the other nice thing about these, the Lumi-804 chelator is when you label or when you just put cold lanthanides like europium in there, you can actually do binding assays. So those are all done at the company. And to my knowledge, there was no difference in affinity of the Lumi-804 antibody constructs than what has been published previously.
Great. So one question would be related to the different elements that you can conjugate onto these chelators. Do you think having identical elements would be important for pairing theranostics, or can we switch between those for imaging and therapy?
Well, that's kind of what we're trying to show with this SBIR is, do we get the same biodistribution when we have zirconium in there versus something like thorium or lutetium? And so we haven't done all the different radiometals yet. And we did show that with two different chelators, you definitely get two different biodistributions. So now we have to go back and look at the metals. I think they're all going to be pretty stable. And I mean, the ideal would be to be able to use zirconium because you get such nice PET images with that, and then swap it out for either a beta emitter or an alpha emitter for therapy.
Right. Great. And maybe I can end with one general question regards to some of Sam's thoughts about moving the bar to the left of staging from advanced disease to detecting and treating early disease. Would you have any comments in terms of how Theranostics may be approaching this question?
To think about that, Theranostics is right now, it's even to get it to be used for early stage prostate cancer like Pluvicto. I'm not even sure. Is it used when it's first diagnosed? I'm not sure. But in terms of but I think where Theranostics is really going to have more of an impact is to be able to first make sure that the patient expresses the particular target that you're looking at so that we can treat people effectively we know that their cancer expresses that target, and therefore we should get higher uptake with the therapeutic. Whether it can be used for early detection, I'm not so sure. I think the diagnostics for sure can detect earlier, and then we can treat. And I think then we can go with maybe the alpha radionuclides for less bulky disease compared to what we can do with the beta.
Right. Great. I agree. I think it's the pairing of diagnostics, in vitro diagnostics, as well as the imaging for late stage. Well, we're on time, so thank you so much, Dr. Anderson. That was a fantastic talk, and thank you for sharing your data with us.
All right. Thank you so much for having me.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.